Welcome to week 2 of Anatomy and Physiology Lab. This week we're going to talk about histology. This week in lab we're going to have our first quiz, and just as a reminder, that's going to cover the material from lab 1 we covered last week, as well as the pre-lab content included in this video for lab 2. So be prepared for that. That'll be right at the start of our lab. Second thing we're going to cover is use of the microscopes. So there is actually a second pre-lab video discussing how to use the microscopes, how to care for the microscopes. And you can also refer to exercise 3 in the lab manual if you need some more information there. And then once we become proficient in the microscopes, we're going to move on to histology, which is lab exercise 6 in the lab manual. And of course, you have a lab workbook that's available on Blackboard, and you'll work through that, complete that during the lab meeting today. Well, the first question to cover today is, what is histology? So as you can see on the slide here, histology is the study of microscopic anatomy of cells and tissues. So for this lab, we'll be using the microscopes to look at these cells and tissues to see what they look like, how they're structured, and how to identify the different cells and tissues. Now, you should recall from lecture that tissues are going to be groups of cells that share both similar structure as well as similar function. And we're going to be looking at the four primary types today, starting with epithelial tissue, also connective tissue, nervous, and muscle tissues. And recall that each of those primary types is going to be further subdivided into multiple subtypes. So we'll have plenty to look at in the microscope today. The first type of tissue we're going to talk about is epithelial tissue. And this is the coatings, the coverings, the linings, basically the surfaces of the body and all the organs. So it's going to be coating pretty much everything in the body. And there are plenty of examples of this, like the skin and the mucous membranes protecting the body from the environment. There's also the linings of the blood vessels and of the hollow organs as well. And what you'll see looking through the microscope is there's a distinct apical surface of epithelial tissue. So it has this free edge. And that is not going to be touching other cells because it's going to be bordering the lumen of these organs. On the opposite side, the basal surface is going to be attaching to other tissues, like connective tissue, for example. So here in this slide, we'll see epithelial tissue bordering that lumen, which is that free space. So the apical surface is the edge that is not connected to any other cells. And then the basal surface at the base of the epithelial tissue is then going to be connected to the connective tissue underlying it. When we talk about epithelial tissue, we're going to want to discuss the naming convention. So there are several types, and they're going to be based on the number of layers of the cells as well as the shape of those cells. So the first part of the name is either going to be simple or stratified based on whether it's just a single layer of cells or whether it's multiple layers of cells. Add to that the general shape of those cells, and it gives you that full name. So simple squamous epithelium is going to be a single layer of these flattened, tapered cells. Cuboidal epithelial tissue is going to be more square-shaped, and columnar is going to be more rectangular. So each of these will have this two-part name. The first, either simple or stratified, depending on whether it's a single or multi-layered tissue. And the second is going to be the shape of those cells in particular. So that'll give us several varieties of epithelial tissue. And we actually have a couple more to discuss as well. These get a little more tricky because, as you can see, with pseudostratified, meaning false stratification, it looks like there are multiple layers of cells. You can see in the diagram over here that the nuclei are layered in different levels of those cells. But if you look closely, the cell membranes extend all the way from the basal surface out to the apical surface. So even though it looks like there might be multi-layers, there is actually only a single layer of epithelial tissue there. So those can be a bit tricky. You can see in this example that oftentimes these will have cilia, so that might help you in your identification, but be sure to pay attention to that in the lab today. The other one that can be tricky is transitional epithelium. And in this case, in the diagram, you can see there are multiple layers of epithelial cells, and you can see some of them are rather cuboidal near that apical surface there. Others are kind of funny shaped. And the key with this transitional epithelium is it actually allows that tissue to stretch. So you'll find this in the bladder in particular, where when the bladder is empty, you'll see it very well multi-layered. And as the bladder fills, it stretches out, it becomes to appear more single-layered. The second tissue type is connective tissue. And connective tissue is this structural and adhesive tissue, basically binds all the other tissues in the body together, gives them strength and gives them support. It's also the most abundant tissue type in the body, and you'll find it everywhere. And as you look through the microscope today in the lab, you'll see that it's a very diverse tissue. So there are a lot of different subtypes, and based on their specific jobs, their specific locations, they can look very different, but they all can be distinguished by high levels of extracellular matrix. And this is that non-living, non-cellular material that fills in the spaces between the cells of the connective tissue. Now, connective tissue is going to be made up of cells as well as extracellular matrix. Now, the cells we see in connective tissue are going to be fibroblasts as well as lymphocytes. The fibroblasts live in the connective tissue. They're specifically part of that connective tissue. Lymphocytes are going to be white blood cells that are commonly found in connective tissue, but may be found in other places in the body as well. And then the extracellular matrix is that non-living, non-cellular material that fills in the spaces between the cells of the connective tissue. And the parts that we'll see is the ground substance as well as fibers. And the ground substance can be a number of things. It can be liquid, as is the case with blood plasma. It could be semi-solid, gelatinous, or even solid, as in the case with bone tissue. And we'll also find in there a number of fibers, either collagen, elastic, or reticular fibers, each of them having different strengths and different properties depending on the specific job of each of the subtype of connective tissue. Now, in general, what you'll find is those fibroblasts inhabiting the connective tissue there. They'll be generating extracellular matrix. You'll also see white blood cells like the macrophage here and the lymphocytes here that will also be found in that connective tissue. And then the three fibers shown here are collagen fibers, which are large, thick fibers, elastic fibers, which are a bit smaller, a bit narrower, and then the smallest are the reticular fibers. And then between all of those structures, you'll find this ground substance filling in the rest of the space. And when we talk about connective tissue, there's several different subtypes, as I mentioned. Connective tissue proper includes areolar, adipose, reticular, and denser fibrous connective tissue. We also include cartilage, bone, and blood as subtypes of connective tissue. Nervous tissue is the third tissue type we're going to talk about, and its primary job is going to be communication. And it has two distinguishing features, which are excitability, or this ability to respond to a stimuli, as well as conductivity, which is the ability to send electrical messages. In nervous tissue, we'll find two subdivisions. And interestingly, although cells are exceedingly small, neurons themselves may be up to 1 meter long. So these neurons are the specialized nervous cells that work to send and receive electrical signals. They're the most widely recognized, the most well-known cells in the nervous system. And you can see them pointed out here on the image of the microscope slide below. It's worth noting that the other subdivision is neuroglia, which are the support cells of the nervous system, and they provide nourishment, support, and insulation for those neurons. Now, while these are not as widely heralded as the neurons, they're very abundant in nervous tissue and have an important job of protecting and supporting and nourishing those neurons. The final tissue type is muscle tissue, and this is going to be responsible for movement throughout the body. There's going to be three subtypes of muscle tissue we'll talk about, but each of these are going to be excitable, so they'll, like neurons, respond to a stimulus. They're also contractile, meaning that once they're excited, they will shorten. They have specialized proteins which will generate contractions and produce force. Now, the first subtype of muscle tissue is skeletal muscle tissue. And as the name implies, it's going to attach to the bones in the body, and it's going to be responsible for our body movements. Now, this is going to be voluntary muscle, so we consciously control its contraction. And if you take a look at the picture of the microscope slide of the skeletal muscle in the corner there, you can see it's very neatly arranged. It has very obvious patterns with striations or those lines running through the tissue. You'll also notice that you'll see a number of nuclei, and actually each muscle cell itself will have several nuclei in skeletal muscle. The second subtype of muscle tissue is cardiac muscle, and this is the involuntary muscle that's found in the heart. Now, this is only going to be found in the heart, so we'll not find it anywhere else in the body, and it is specifically set up to cause those contractions to generate pressure on the blood in the heart. What you can see from the picture here is that there's a lot of similarities to skeletal muscle. So you will see a somewhat parallel arrangement here, although there is a little bit of branching involved, so it's not quite as neatly arranged as skeletal muscle, but there are still the obvious striations running across that tissue. A couple important differences, though, is typically cardiac muscle will be uninucleated, so there is only one nucleus per cell. You can also see the structures called intercalated discs, which form junctions between those cardiac muscle cells. Now, the final subdivision of muscle tissue is going to be smooth muscle. This is, again, an involuntary muscle, so similar to cardiac in that respect, and it is found throughout the internal organs. So this is, again, going to be subconsciously controlled, and in this case, it actually looks quite a bit different from the other two subtypes of muscle tissue. So smooth muscle, as the name implies, does not have the striations. It has a smooth appearance to it. It also has these spindle-shaped cells, which are kind of flattened and short, so quite a bit different from what you'd see in cardiac or skeletal muscle. It's also, like cardiac muscle, going to have a single nucleus, so it is similar in that way, but it is fairly distinct from the other two types in some of those other aspects. As we wrap up with the pre-lab video here, a couple things to keep in mind for this week's lab is that, one, there is a whole lot of material, probably a lot of new material for you, and it tends to be fairly difficult for students. So we want to make sure you're making use of your resources. With your time in lab, make sure you're looking at the microscopes, looking at those different slides, being able to see and identify the different tissue types. Also recall you have those images in your lab manual. You can also find them online on a Google image search or a number of resources there. Make sure when you see those slides, you know what you're looking at, and you could tell me which subtype of tissue that is. And when you inevitably have questions, make use of your professor, make use of your TA in lab. They'll be able to help you work through the different slides and how you can identify all of these tissues. Because at the end of the day, you're going to have to recognize and identify these different tissue types. If you're shown a slide in a quiz or in an exam, you're going to have to be able to tell me what tissue type is it, what subtype is it, so make sure you're comfortable with that. And finally, take your time. 3 hours seems like a long time in lab, but it'll go by pretty quickly. There's going to be a lot to go over. So take a breather, take your time, make sure you're making use of, especially the microscopes and the slides, which you probably don't have at home. So you only have a limited amount of time to look at those. So that's one of your resources. Make sure you use that wisely, and we'll be in great shape for next week's quiz and the midterm in several weeks. Thank you.
Hi, this is Dr. Crosswhite. Today I'm going to talk about the integumentary system, or more commonly referred to as our skin. I've listed the learning objectives for this exercise. They're also on page 93 in your lab manual. A good idea after listening to this lecture, or when you're studying for an exam, is to go through the learning objectives as a review method. I'm not going to read them off right now, but these are the concepts and the facts that you should know when you're finished listening. So today I'm going to briefly talk about the basic function that the integumentary system provides, but the majority of this lecture is going to focus on the structure of the skin and the accessory organs. Okay, so let's begin. The integument, or skin, is considered an organ. In fact, it's the largest of our organs if we're talking about the percentage body weight. And it's a very fascinating organ because it provides so many different functions to us as organisms. The first thing that you're probably aware of is that the skin is pliable or flexible, but at the same time it's very tough. So this enables our skin to withstand, to a certain degree, many different abrasions or other threats to it. Our skin also has many other functions. It insulates or cushions the underlying body tissue, so it's protective again. It helps us prevent too much water loss or dehydration. It plays an important role in regulating body temperature or thermoregulation. It also acts as an excretory system, as we have all experienced body sweat at one time or another. Our skin is also an important site for vitamin D synthesis, and vitamin D is critical for calcium absorption in the digestive system, so it does play important metabolic duties. And finally, our skin is a critical site for the many sensory organs, including touch, pain, pressure, and temperature. So these are the basic functions, but let's move on and talk about the basic structure of the skin. The skin has two major layers, the epidermis and the dermis. The epidermis is the outermost or superficial layer, and it's relatively thin compared to the dermis. This is figure 7.1 from your book, showing the epidermis right here and the dermis layer here. The epidermis is composed mostly of epithelial cells, and it is divided into 4 sublayers. 5 if we're talking about thick skin, such as the skin on our palms or feet. The dermis lies below the epidermis. It's composed of various connective tissue, and it is also divided into 2 subregions, and this is the site of where most accessory organs of the integumentary system are located. So let's look at each layer in more detail, starting with the epidermis. Figure 7.2 shows a magnified image of the epidermis. I'm not going to go into too much detail regarding what is happening at each of these layers. That information can be found on table 7.1 on page 96 in your lab manual. So the outermost layer, or the one exposed to the outside environment, is the stratum corneum. And this layer can vary in thickness. It's composed of dead, scale-like keratinocytes, and these are the skin cells that we continuously are shedding and replacing. The layer below that is the stratum granulosum, and this contains granules that produce a glycolipid, which provides our skin with its waterproofing ability. The layer below that, which is the largest layer, is the stratum spinosum. And underneath the stratum spinosum is the deepest layer, the stratum basale. And it is a single layer right above the dermis, and the cells at this layer are constantly undergoing mitosis to generate new cells. Also in this layer, about 10 to 25% of the cells are melanocytes, which I will explain on the next slide. And then not shown in this figure, there is another layer that occurs in thick skin regions like our palms or bottoms of our feet, and that is called the stratum lucidium, and that is shown on figure 7.6. So let's talk about the cells, the types of cells that are located in the epidermis. Keratinocytes are the most abundant cells present, and they produce keratin fibrils that provide skin with its durability and protective capabilities. The keratinocytes are being produced at the stratum basale, so down here at the lower levels of the epidermis. And these cells migrate up and eventually die, and we shed them off. So therefore, the stratum corneum is composed mostly of dead keratinocytes. Melanocytes are pigment-producing cells, and they provide the skin with its color. The melanin pigment that is produced by the melanocytes also provides protection to keratinocytes and other cells underneath. The skin also contains dendritic cells, which are a type of immune cell, and these are important cells for phagocytosis of potential bacterial or viral threats. And then lastly, there are tactile or Merkel cells, which are part of our touch sensation. These cells lie at the epidermal-dermal junction, and these are the main types of cells that are located in the upper layer of the integument or the epidermis. So let's move on and talk about the layer below, the dermis. The dermis is a much larger layer than the epidermis, and it's composed of mostly connective tissue, including collagens and elastins. And these are what provide the skin with our youthful appearance, and as we age, the number of elastic fibers decrease, causing our skin to wrinkle. The dermis is a very important site for the accessory organs, including hairs and nervous system structures. But the dermis is divided into two subregions, the papillary and reticular layers. Notice that the papillary layer is very thin compared to the reticular layer. Very quickly, I want to highlight the main functions that the dermis provides. It provides the elasticity to the skin that I mentioned previously from the collagen and elastin fibers produced. It also allows us to radiate excess heat to the outside environment through the capillary networks located here, and it provides our skin with various sensory organs necessary for touch, temperature, pain, etc. So let's talk about both the papillary and the reticular layer in a little bit more detail. Papillary layer is the smaller upper region of the dermis, and it lies just beneath the epidermis. It can vary in thickness, but it's attached to the epidermis via dermal papillae, shown here, which on the hands, the dermal papillae, they produce the fingerprints, or the unique patterns of epidermal ridges that remain unchanged throughout life. The papillary layer is highly vascularized with capillaries, which not only provides nutrients to the epidermis, but it also allows us to radiate excess heat to the skin surface. Here is where you can see the capillary layers, the capillaries that are located in the papillary layer. Finally, there are many free nerve endings for pain sensation and touch receptors that are located in the papillary layer. The reticular layer of the dermis is the deepest layer in the integument system. Its depth can vary depending on the amount of connective tissue and fibers. It is also highly vascularized and contains many additional structures, including sweat and sebaceous glands. Sebaceous glands shown here, sweat glands shown here, and which we're going to go over in a little bit more detail in a few minutes. Also, the reticular layer contains hair follicles and pressure receptors. Also, I want to mention that the hypodermis lies below the dermis, which is shown here, but it's not considered part of the integument. It is composed mostly of adipose tissue. So that's the basics of the epidermal and dermal layers of the integument. Let's move on to discuss the various accessory organs in the integument and the roles that they provide. There are three main types of accessory organs in the integument: nails, hairs, and cutaneous glands, which can be sebaceous or sweat glands. All these structures, they reside in the dermis, but they originate from the epidermis. Let's take a look at nails first. Nails are horn-like structures that originate from the epidermis. They contain many parts, as shown in this figure. The body is the largest part of nails. The free edge is the part that we are constantly or regularly clipping with nail trimmers. The hyponychium is the region underneath or on the underside of the free edge. The root is the part of the nail that's embedded in the skin, and it is anchored by attaching to an epithelial nail bed, shown here. The nail folds overlap the borders of the nail, and the eponychium, or more commonly called the cuticle, is a projection of the thick proximal end of the nail fold. The nail matrix contains germinal cells, which are responsible for continuous nail growth. As the matrix produces nail cells, they become heavily keratinized and they die. So nails, like our hair, are mostly composed of non-living material. The last structure that I want to mention is the lunule. It is the proximal region of the nail matrix, and it appears as a whitish color. Everywhere else on our nail, they appear as pink, but they're actually colorless, and they appear pink due to the blood supply that is located in the underlying dermis. So that's the basics of nails, and let's talk about hairs next. The second accessory organ of the skin is hair. Hairs are enclosed within follicles. They're found all over the entire body surface except for thick-skinned areas mentioned previously, but also areas such as our lips, nipples, and parts of the external genitalia generally lack hair. There are two main regions for every hair. The shaft, which is the part that extends above the skin, shown here, and the root, which is located below the skin and is enclosed within a follicle. In addition, each follicle has a small muscle that connects it to the papillary layer. These muscles are called erector pili, and when they're contracted, the normally slanted hair follicle is pulled upright. We all experience this when we get goosebumps. Each hair has 3 layers, shown in this top figure here. The innermost medulla, and then each hair has a cortex, and then it has this protective cuticle area. If the cuticle is damaged from abrasion, this is what results in what's called split ends. Each hair follicle has a protective sheath, and it's composed of external and internal epithelium. At the root, each follicle contains a small nipple of dermal tissue that extends into the bulb to provide nutrition to the growing hair, and this is called the papilla, shown here. At the top of the hair papilla, there's a thin layer of actively dividing epithelial cells called the hair matrix. And these cells provide the follicle with actively growing hair cells. Finally, there are melanocytes that are located in the follicle, and they're located near the papilla, and our hair color depends on not only the amount but also the type of melanin pigment that is produced by these melanocytes. The final type of accessory organs to discuss are the cutaneous glands, which fall primarily into two broad categories: sebaceous glands and sweat glands. Sebaceous glands, which we're going to talk about first, are found nearly all over the skin, with the palms and the soles of the feet being the only exception. The ducts that are associated with these sebaceous glands usually empty into a hair follicle, as pictured here and on the next slide. But some actually open directly onto the skin surface, so it can be either or. Sebum is the product produced by sebaceous glands, and this is a mixture of oily substances and fragmented cells, and it acts as a natural skin lubricant to keep our skin soft and moist, but sebum also helps prevent hairs from becoming brittle. Sebaceous glands are particularly active during puberty when increased amounts of androgen hormones are being produced, and this results in the skin to be a little bit more oily. Also, something that we most all have experienced are infections of sebaceous glands. So when a sebaceous gland becomes infected with bacteria, you have acne. And blackheads, which are a type of acne, these are accumulations of dried sebum, but it also contain bacteria and the melanin pigment from epithelial cells in the oil duct. This picture is just illustrating a sebaceous gland, shown here, where the duct is emptying into a hair follicle. The second type of cutaneous gland is a sweat gland. Again, they are distributed widely over the skin, and their outlets are called pores, shown here. Sweat glands are categorized by the composition of their secretions. There are two types, eccrine and apocrine. Eccrine sweat glands, pictured here, produce clear perspiration, which is mainly composed of water, salts, and urea. Eccrine glands are under the control of the nervous system, which explains why people tend to sweat more when they're nervous or when they're excited, despite the temperature of their environment. Eccrine sweat glands are also important for our ability to regulate body heat. They secrete perspiration when the external temperature or internal body temperature is high. As the perspiration evaporates on the skin surface, it releases the excess heat, and the evaporation also cools the surface of the skin and the underlying blood in the capillaries. It cools the underlying blood in the capillaries, which helps lower our core body temperature. Apocrine sweat glands, which are not pictured in the lab manual, they are found predominantly in the axillary or arm regions and the genital areas. Apocrine sweat glands secrete the same components of eccrine glands, but they also secrete proteins and fat-rich substances. These glands are responsible for producing our body odor, which occurs when bacteria on our skin surface break down the organic secretions, which produces the often unpleasant smell associated with body odor. So that finishes up the integumentary system for today. Don't forget to review the learning objectives on page 93 in your lab manual. We covered basically the functions of the skin, its main components, their anatomy, as well as the role of the accessory organs associated with the integumentary system. That's it.